Hello biologists, this is Lab 3.4, Digestion Enzymes, and we're going to do the bonus question at the end of your lab for 10 extra credit points. The question that we're asking here is, what are some other enzymes that might be able to digest the protein in eggs? What are some real-life enzymes that we might encounter that could digest protein? Well, we know one enzyme that we talked about in this lab that you can readily find in real life. It's lactate. But we know lactate digests lactose, which is a sugar. Now eggs, as you know, are mostly protein. So enzymes are pretty specific, and it'll be interesting to see that if this enzyme that's meant to digest a sugar can digest a protein. Bromelain is a protease. It's the stuff you find in meat tenderizer. If you look very carefully on the back of your meat tenderizer, if you have any, it contains an enzyme called bromelain, which is found in pineapples. And it will kind of break down your steak. And since steak is mostly protein, this is probably a pretty good candidate. The optimum temperature for maximum activity on this enzyme, as we know enzymes are very pH and temperature sensitive, this one likes 35 to 45 degrees Celsius. Well, it doesn't like. Enzymes are not people, they don't have feelings, but it works best at 35 to 45 degrees Celsius. Now, papain is another real-life enzyme that you can buy in most health food stores. It's from papaya. It also digests proteins, and in fact, if you have a steak in some South American countries, you get a lovely dessert afterwards that's made from papaya because it'll help you digest all that steak. Papain is the name of the enzyme, and papain, of course, as all enzymes are, is temperature sensitive. It has an optimum range of 60 to 70 degrees Celsius. That's pretty darn warm when you consider a warm, very hot summer day is about 40 degrees Celsius. Another candidate here is alpha-galactosidase, or what you can find in Beano. Now, Beano is found in you in limited amounts, and it acts on ribose. Ribose is a trisaccharide. That's a 3 sugar. It has 3 sugar groups here. It's found in things like cabbage, broccoli, and beans. It's the thing that your body has a hard time breaking down. And so some people take Beano so they can break that sugar down and not feed the bacteria in their lower intestine that causes gas. So at this point, let's think about our real live enzymes, where you find it in real life, and then predict, will it digest the protein in eggs or not? And this table is on your lab sheet that you'll find in doc sharing. Lactase digests lactose. You'll find it in real life in Lactaid or other lactase containing products. Will it digest the protein in eggs? Well, you remember lactose is a sugar found in milk. Bromelain is a protease. That is a protein digesting enzyme. You find it in meat tenderizer, and they got it, the meat tenderizer folks got it from pineapples. Will it digest protein? Well, it'll help digest the collagen, the tough stuff in your steak. Collagen's a protein that's also in your connective tissue besides a cow's or a pig's, depending on what kind of steak you're having. Collagen is the stuff in your nose, in your ears. It will digest that and make your steak more tender. Papain, remember, is a protease as well. It's a protein enzyme. It's found in papaya in real life and extracted into tablets. Will it digest protein? Well, it is a protease. Galactosidase digests ribinose, which is a trisaccharide in cabbages, broccoli, and carrots. You find it in real life in U and in Beano. And Beano gets its alpha-galactosidase from a species of fungus that also uses the same enzyme. Will it digest proteins? Well, remember, ribinose is a sugar. It's a trisaccharide. So will something that's meant to break down a trisaccharide, a sugar, break down a protein? Well, we'll find out. Let's look at the results. Before we talk about too much of the results and I show you how much of the eggs were digested, I let things sit, the enzyme, the eggs, sit in a container with a heating pad for about 36 hours. And since many of these enzymes work best in a range that's body temperature or greater, I kept them nice and toasty with the heating pad. Before I show the results of any of the enzymes, this is what eggs look like. I cooked some eggs. I had the egg white smashed up, and I put a tablespoon of egg white in a test tube. And both of these had no enzyme. I know enzymes are pH sensitive, so I put some eggs. So in my experiment here, I used acid and base, vinegar and baking soda, to create acidic and basic conditions, but I wanted to make sure that the acid and base weren't going to digest the eggs. So this is egg in vinegar, and this is egg in baking soda. Unfortunately, my egg in water didn't survive very well. So you can see that both of these are pretty much exactly the same. The eggs still look fluffy and just like they did when they went into the test tube. So these are our controls. Here's the vinegar, here's the baking soda, and there was one in water as well, and it pretty much looks exactly the same. So here's our first set of results. This is papain. Papaya is kind of an orangish color, so is papain. And here is the eggs in vinegar. This is papain in vinegar. This is the eggs in vinegar. So you can see that just plain eggs are still nice and fluffy, and the eggs that had the papain in them are kind of sunk down inside the test tube. So papain in vinegar compared with just eggs in vinegar, it looks like there was quite a bit of digestion here, which makes sense because papain is a protease, and proteases break down protein. So we saw quite a bit of digestion right here. Now let's compare papain, the orange one, to just papain in water to eggs in just water. Now papain apparently likes a little bit of an acidic environment to work in, and because these are pretty much the same. So not a whole lot of digestion in just plain water, whether you just have the control or the papain. Results are kind of inconclusive when we compare the papain in baking soda with just plain eggs and water in baking soda. So you can put inconclusive on this one. There looks like there might have been some digestion, but there's some funky color changes too. I would want to do this experiment over again if I wanted definite results. Real science sometimes takes a lot of work, and sometimes you have to redo your experiment, and that's what happens. That's real life. So we're right here, Beano in water. Here's the control. It's just eggs in water, no enzyme. Or actually, I got them mixed up because they're so similar. Here is the control, and here is the water in Beano. And they're, pretty much similar. So I wouldn't say that Beano in water did a whole lot. Here's Beano in vinegar, this one. That looks pretty digested, surprisingly, even though Beano is supposed to act on a sugar. It looks like it did okay at digesting the egg, because this is a control. This is just egg and vinegar. So even though Beano is meant to work on a sugar, maybe it works a little bit on the egg as well. And here's baking soda, a base, and Beano, and here's just water and baking soda, no enzyme. So these are pretty much the same. The control and the Beano, just the water and baking soda, and the Beano water and baking soda are pretty much the same. So I don't think Beano works very well in a basic environment, which makes a lot of sense because you remember that your stomach's pretty acidic. And if you're going to swallow Beano and expect it to work, the enzyme has to survive and not denature, not fall apart, not lose all those important connections at pH 2. So now we go down to the bromelain or meat tenderizer in water. Here's just the meat tenderizer and water, no pH adjustments, pure pH 7. And here's just eggs and water, no enzymes. And you can see that even at regular pH 7, the bromelain digested the eggs a little bit. I think this may be the clearest winner here. Here's meat tenderizer in vinegar, working at an acidic environment. And here's just eggs in vinegar with no enzyme. You can see that there's a very big difference. If you put the meat tenderizer in there, you get a whole bunch of digestion. The eggs kind of get mushy and settle down to the bottom of the container. And if you just put the eggs in vinegar and let them sit, nothing really happens. In fact, that's how you make pickled eggs, is you just put eggs in vinegar. So obviously the meat tenderizer did its job here and the enzyme digested some of those proteins. Remember, bromelain is a protease. It's a protein digesting enzyme, and so it should work pretty well. And if it works in your stomach, it doesn't have to. You can tenderize meat anywhere, but apparently it does okay in an acidic environment. Not every enzyme does. Some of them need a more basic environment. As it turns out, it looks like bromelain does okay in a basic environment here. You can see that it digested the enzyme digested the eggs a little bit. Here are just regular eggs and water and baking soda, no enzyme. Here's the one with the enzyme, here's the one without. You can see that it digested it a little bit in baking soda, not as well as it did in the vinegar. So enzymes are pretty pH sensitive. Some work better at low pH, some work better at high pH in a basic environment, and they're also temperature sensitive. Some work at low temperatures and some work at high temperatures. I'm going to let you make up your mind about which one you think it works best in. Here's the meat tenderizer in vinegar. Here's the meat tenderizer in baking soda, a base. Here's an acid, here's a base. Which one do you think meat tenderizer works better in? You can make up your mind. It works a little bit in both, but it seems to me like it works much better in this one, which is a low pH environment. It's got vinegar in there, it's acidic. Our last enzyme is lactase. Remember, that's supposed to work on lactose, which is a milk sugar. Well, we only have one comparison here. We have vinegar and vinegar. This one has lactose in it, and this one doesn't. So there's a little bit of difference, but not surprisingly, an enzyme that's designed to break down a sugar doesn't work very well to break down a protein. And unfortunately, these two test tubes did not make it. So we'll have to leave those blank. But hopefully, because of this lab, you realize that enzymes are very specific. They only act on a particular kind of molecule, and that they're sensitive to things like temperature and pH. Lactase works best in your small intestine in pH about 6. Your body adds some sodium bicarbonate to the contents of your stomach in order to neutralize it and bring it down to a more neutral pH. Remember, here's the pH scale, and pH 6 is about right here. pH 2 is where your stomach is, about right there. Bromelain is a protease found in pineapple, works great on proteins, and we found, and it also